Earlier this semester, we learned about how DNA has to copy itself during interphase of mitosis. Hopefully you guys recognize this diagram as the steps of mitosis, and that being cell replication. Now remember that it's very important to get exact copies of DNA when replicating it, because you really don't want to have any mess-ups when it comes to DNA. This is an overall image of what DNA replication looks like. Now let's think back to the model that we've done in class of DNA structure, and if we remember from our model, DNA runs in opposite directions. So if we look at this, we're going to see how that works with this end of DNA up here being the 3' end, and this end of the DNA being the 5' end. And since they run in opposite directions, remember that this will be 5' down here and 3' over here for these opposite sides of DNA strands. Okay, and we're referring to the backbone of the ladder when we talk about it. And remember, the backbone is made up of those deoxyribose and phosphates. So let's just clear that off, and we will look at how DNA replicates itself. So the first step to DNA replication is going to be this enzyme here, going right down the middle, unzipping DNA. So step number 1 is going to be that helicase unzips DNA right down the middle. So now we've got these free nucleotides, just they're no longer bound together with their opposing base pair. So the first part that happens is we are going to have this really cool enzyme called DNA polymerase go through and work in specific directions, and it only can work in 3' to 5' direction. That's very important when it comes to DNA replication, and you're about to see why. So this DNA polymerase is working in a 3' to 5' direction, adding these free nucleotides that are just kind of floating around, and it recognizes that, and it's actually able to grab on and add those free nucleotides to the open DNA strand. And when it's really easy to do this and the DNA is working on this, it's called the leading strand. So then we come down to the opposite strand, right, where it was down here and it was going 5', 3' on this end. Well, we've got a very similar DNA polymerase right here, you can see it. But remember, I said it can only work 3' to 5'. This is very important. So since it can only work 3' to 5', it's got to have a way to do this. And the way that it gets around this is that this DNA polymerase is going to go from the 3' end to the 5' end, and then when it gets to the end where it's already been unzipped, it's going to jump back to where the helicase is still unzipping and follow along. Then it's going to jump back, follow along, jump back, follow along, always working in the 3' to 5' direction. And when it's doing this, it's actually making the DNA, adding the DNA in little fragments, and we call these fragments Okazaki fragments. And it's called Okazaki because the gentleman that discovered these fragments and discovered how this DNA polymerase only worked in one direction, his last name was Okazaki. So let's review really quickly. We've had our helicase unzip our DNA right down the middle. Step number 2 is DNA polymerase is going to add free nucleotides to the unzipped DNA. And then another DNA polymerase is going to work on the lagging strand by adding Okazaki fragments. So now the problem with these fragments up here, though, is they actually aren't connected all the time. And so in order to connect them, we are going to use another enzyme called ligase, glues them together. So one more time. Helicase unzips the DNA, and then DNA polymerase up here on the leading strand works in the 3' to 5' direction, adding these free nucleotides onto that open DNA. And then on the lagging strand, the DNA polymerase adds free nucleotides in 3' to 5', but only does it in little fragments called Okazaki fragments. Now, in this picture, this DNA polymerase is going to keep on going until it runs out of DNA over here, and then as soon as it runs out of DNA, it's going to pop off and go back to the start up here and add new DNA. So what we call this is actually called semi-conservative replication. Let me write that out for you. Semi-conservative. I'm going to run out of room here. Semi-conservative replication. And that's because when you end up with this new DNA, here's your original strand of DNA, your original template. It splits up the middle. You start to add these new nucleotides. And then on your two new strands, you've got old strands here. And then you've also got these newly synthesized, newly made strands of DNA. So you're always going to have 2 of the old and 2 of the new. I hope that this helps you to understand DNA replication, and let's model this in the classroom today.